Assalamualaikum and a very good day to everyone. Today our Group 5A will proudly present to all of you a video entitled Amniocyte is a subclass of extraembryonic cell. It is a cell of a fetus which is suspended in the amniotic fluid. Amniocytes are considered to arise from several tissues including fetal skin, the fetal urinary tract, umbilical cord, and inner amniotic surface. Amniocytes include amnion-mesenchymal stem cell, amnioserosal cell, amniotic epithelial cell, chorionic membrane mesenchymal stem cell, and fibroblasts of villus mesenchyme. Now we'll be moving on to method of amniotic cell culture. To grow amniocytes, there are two ways of culture based on the cell types we need, either adherent or suspension culture. The sample is obtained from the mother through amniocentesis. Amniocentesis is a procedure in which amniotic fluid is removed from the uterus for testing or treatment. Next, the amniotic fluid is centrifuged. After the centrifugation, there will be two layers in the tube, which are the pellet and the supernatant. To culture amniocytes which are adherent cells, we extract the pellet for the following steps. On the other hand, if the amniocytes of suspension type is needed, then we will take the supernatant instead of the pellet. Now, we begin with the culture of amniocytes of adherent type first. Pellet is obtained from the tube and is resuspended in the culture medium. Then, the solution is seeded onto a culture flask. When they are seeded on the flask, they are brought to incubate at 37 degrees Celsius for 5 to 7 days to allow for proliferation of the cells. After this, the condition of amniocytes of the primary culture can be assessed under the inverted microscope. Once the cells are cultured, they need constant maintenance such as changing medium and preventing toxic condition in the flask. Amniocytes morphology under the microscope could tell whether the cells cultured are healthy or not. The rounding up of the cells shows that the cells require new medium as the old medium has been used up. The adherent cells can be subcultured to increase the number of cells. To subculture adherent amniocytes, the old medium has to be removed. TE buffer is added to wash cells. Buffer washes away the traces of medium to ensure the purity of the culture. Trypsin is added to detach the adherent cells. Then, the amniocytes are incubated in order to detach cells completely. After that, the cells are checked under microscope to make sure all the cells have been detached. Moving on to the culture of suspension amniocytes, the supernatant, which contains the suspension cells in the tube instead of the pellet, is obtained after the centrifugation of the amniotic fluid sample. The supernatant is seeded onto a flask with a new medium added with fetal bovine serum (FBS), allowing the suspended cells to grow. However, always bear in mind to practice the aseptic techniques and the proper use of laboratory equipment when working with cell cultures. Always use sterile equipment and reagents, and wash hands, reagent bottles, and work surfaces with biocide or 70% ethanol before beginning work. You might be wondering what are the applications of the cultured amniocytes. One of the applications is that the cultured amniocytes can be used for the diagnosis of trisomy 21 at around 12 to 18 weeks of pregnancy using amniocentesis technique. However, the diagnosis includes a method called chromosome preparation, and here are the steps involved.
Shoelaces, yeah, over my shoelaces. Got to get a new pair of shoes to kick it with her, not kick it with you. Got to get a new pair of shoes to kick it with her, not kick it with you. Second time broke out a bone, I tripped over my shoelaces. Second time broke out a bone, I tripped over my shoelaces. Second time broke out a bone, I tripped over my shoelaces. 100-meter races, yeah, 100-meter race. Got to get a new pair of shoes to kick it with her, not kick it with you. Got to get a new pair of shoes to kick it with her, not kick it with you. What she gon' do like you? 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 What she gon' suck to me? Toe bone into the ankle bone into the shin bone into the knee bone into the thigh bone into the hip bone into the backbone into the collarbone into the neck bone into the neck bone into the collarbone into the backbone into the hip bone. If wanna gummy matty, on them got me, and when you go